All right, I wanted to briefly talk about the respiration lab and refresh your memory. This is the lab that we did with the germinating peas, and we put them in these respirometers, which are glass tubes. Um, then we measured the amount of either CO2 produced or oxygen consumed, which is the same thing, with the water movement in the glass tube, in the pipette, essentially. So we used the peas as a respiring organism that consumes CO2. I'm sorry, that produces CO2 and consumes oxygen. We should not be looking at the peas as photosynthetic plants at this point. There isn't any photosynthesis going on because the peas are at a place in their life cycle um, where they're using the stored up energy in the seed itself, sort of to get the plant going, to start growth. And then eventually, once leaves grow and everything, photosynthesis will start. But that hasn't happened yet. So we're purely strictly looking at respiration, and the green color of the peas shouldn't be something that should confuse you here. When we take a look at the chemical equation for photosynthesis, which can be seen right here, we see that it's a glucose, and 6O2 will produce 6CO2 and 6H2O. That's the balanced equation. The dilemma is that if you decide to uh, look at the oxygen consumed, the problem is that for every uh, volume of oxygen that's consumed, the same volume of CO2 is produced. So stoichiometrically, these two are equal. For every 6 moles of oxygen, 6 moles of CO2 is produced. Um, so that's an issue if you're measuring, a volume in a container with this pipette. If you're looking at the pipette tip and you're looking at the water in here moving either, inward or outward to try to see if the volume decreased, whatever O2 was consumed and decreased the volume in the setup was then replaced by CO2, which keeps this in the same place. So we solved that problem with KOH. So KOH is a chemical that reacts with CO2 and takes the CO2 out of the situation and um, causes it to precipitate and then dissolve. So it's no longer a gas, and the entire volume of that CO2 disappears then, and essentially the volume inside of the respirometer shrinks as the oxygen gets consumed. There is no gaseous CO2 then remaining at the end because KOH takes it out of the system. So that's a good way to actually allow this to move and actually be an indicator of oxygen consumed. So we can measure on the pipette then, how many milliliters, it's fractions of a milliliter. So the entire pipette length was 1 ml. So if it moved to, 1 mark in, that would have been at zero point 0.1 ml, and anything beyond that is 0.01 ml, the smaller increments. So what we did is we set this up by placing these respirometers in water, and then we allowed them to sort of equalize in terms of temperature and all of that. We had to let them sit in there for quite a while to allow the temperature to become the same, and that's really, really important because if you put these respirometers in a bath of water and the water is colder than the air temperature or than the respirometer, to start with, the cold temperature in here will cause this volume to decrease and then draw the water level in, which is an artifact. That's fake. It's a fake result. So basically, we allowed them to sit in there and equalize, and that then um, prevented that kind of a you know faulty type of data. Um, we did not do dry peas. We just skipped this because to me this is kind of a foolish part of it. We don't really need to worry about that. We did glass beads and we did actually live germinating peas. And the glass beads acted as a control. So ideally, if everything is equalized perfectly, the water level in the glass bead system shouldn't move at all. There should be no respiration, there should be no shrinking or anything like that of the volume. So that should stay in exactly the same place. But if there's some kind of a change in temperature or change in pressure in the room, if somebody opens a door or something like that, and this moves a tiny little bit, so that let's say this thing moves by 0.02 mLs, and then this thing, the actual one that you're measuring with the live peas, moves by, let's say, 0.20, You just simply subtract out this movement from this movement. So you would say this is 0.18. That's to sort of, adjust for any kind of error or any kind of fluctuation that's happening with the environment. The actual movement just due to respiration would be 0.18, not zero point 0.2, because we saw this one with the dead glass beads also moved by 0.02. Um, so that's something to consider. And the other thing we did then is we compared cold temperature versus room temperature, and we just saw that at room temperature they had good rate of respiration, and that at a cold temperature there was a lot less, or maybe this was even further down. And then the glass beads should have been, like down here somewhere really low. Sorry, badly drawn graph. The dry ones also should be really low because they really have minimal respiration going on. I think that about sums up this one. Just kind of look this over again and think it through just to make sure you understand all the components to it. Why did we put them in water? Um, why did we let them sit? What does each of the, what do the measurements mean? What's the point of the KOH? All of that stuff. And then, scientific method. What's the point of the control? Why do we have to have a control and all that? So scientific method seems to be a real trendy thing for them to be asking about, and I wouldn't be surprised if that showed up on the test. So always keep that in mind when you're thinking about these experiments.